Hi everyone, sorry I cannot meet you in person due to COVID. My name is Sebastian Sensale. I'm a postdoctoral fellow at Duke University, and to tell you about the work me and my collaborators have done related to how to optimize communication speeds in the Origami devices. So, DNA nanotechnology is a field of research invented by Ned Seaman from NYU in the early 80s, which consists of making complex structures out of DNA. Now, to make a DNA origami structure, we start from a long single-stranded molecule of viral DNA, which we call scaffold, and we fold the structure by adding stable strands designed to bind at strategically defined locations. This results in the formation of a predefined 2 or 3-dimensional shape. The design of these shapes is purely computational, and we use specialized software such as Nano or Magic DNA for this task. So what are the uses of DNA origami structures, and why do we want to use DNA as a building material for nanostructures? So the first and most important characteristic of DNA is that it provides real nanometric precision. We have control of every single nucleotide of these structures, and we can make crossovers between helices or place individual nanostructures every 2 nanometers or 7 base pairs. We can also build structures with very high yield and in a very reproducible way. Another cool characteristic of DNA origamis is their biocompatibility. DNA origami structures are non-toxic, they're relatively chemically inert, and we can tune their degradation time, so they're fantastic candidates for drug delivery. However, they're immunogenic, that is, they can produce an immune response, so we really need to be careful and to understand the sequences that we are using in our molecular designs for in vivo applications. The third feature is that DNA origami structures are actually programmable. We can program motion of 2D and 3D DNA origamis as a response to different stimulus. Things like molecular bindings, changes in temperature, in salt or hydrogen concentrations, as well as electric and magnetic fields. To me, this is very interesting, and one of the things we work with in Arias Lab is the computational modeling of dynamic DNA origamis. Now, there are many challenges in the computational modeling of these devices. To start with, the characteristic length and timescales make it impossible to study this phenomenon with all atomic precision. In fact, even existing coarse-grained models, such as OXDNA, cannot model these actuation dynamics. So we must recur to other modeling strategies. Now I'll give you a few examples of the techniques we have developed in our lab. So probably the easiest example to study from a fundamental perspective is that of communication in DNA origami devices. So let's say we trigger a conformational change in one part of a DNA origami designed to set off a domino effect. And we do this, for example, by having our trigger molecule respond to a binding reaction. What we want to know is how long will it take to propagate a response to a stimulus, to propagate the signal, and how can we optimize our design to enhance the speed of propagation. So to this date, there's nothing that moves like a domino made of DNA origami, but the DNA acrobats designed by Hao Yan and Niels Waters groups from Arizona State University and U Michigan, they move close enough. So this device consists of a DNA origami platform with tethered molecules on top, which diffuse around their pivots. So here you have a simplified diagram in 1D. Imagine that these things can move in plane and out of plane as well. Now, one of these DNAs is different than the others. It's double-stranded because it carries a signal. It carries a single strand in the molecule with a protruding section complementary to a foothold of its neighbor. Now, this protruding section, the short single-stranded overhang here, is called a toehold. And when this toehold binds to the base of its neighbor, it can jump from one strand to the other through a sort of random walk that we call branch migration. Now, branch migration is not always successful, and the probability it will happen depends on the properties of the strand and of the toehold itself. But when it is successful, the signal can jump to a new strand, and this signal, this walker, can go back and forth between neighbors. So if we attach a fluorophore to the signal, to this walker, and look from the upside, we're seeing an effective diffusive motion center at the initial position of this walker. And tracking the square displacement of the fluorophore over time, we can estimate an effective propagation speed. While by means of FRET, we can get the real communication time between one neighbor and the other. You can probably imagine that the spacing will come into play in these dynamics. If we separate the DNAs to match, it will be impossible to propagate a signal, unless we increase the length of these molecules. So there has to be an optimal relation between all these parameters. And in Arias Lab, we developed a theory for this. So we start by observing that for a double-stranded DNA molecule, a molecule much smaller than its persistence length, the free end of the molecule moves along the surface of a hemisphere. And this motion can then be mapped to the motion of a particle diffusing a two-dimensional potential, which in this case was actually very well approximated by a harmonic potential. Now, to cursorize binding, we first observe that the probability of finding our particles at a certain location xy is related to the potential fields by means of the Boltzmann distribution. Now, changing our system of coordinates, changing the origin of our system of coordinates to a target, and performing an orientational average, we can explicitly obtain the probability density associated to finding our particles and our target at a certain distance ρ. So if we consider a walker that moves in 1D as the distance between particle and target, as ρ, which feels an effective potential associated to this probability density, here you have the relation, then, via the Smoluchowski equation, we can derive an expression for the time it takes for this walker to bind to our target, if both walker and target can bind when they are at a distance of epsilon. Of course, if epsilon is 0, this time will be infinity, as it is quite hard to play golf with an infinitely small hole, even if we have an infinitely small ball. So let's assume epsilon is greater than 0. What we have right here, then, this expression for the mean binding time is exactly what we wanted. And it consists of two terms. We have one term which represents the time it takes for the first encounter between particle and target to happen, and one term that represents the time it takes for eventual reflections to lead to a successful binding event. Because remember what I told you before, branch migration is not always successful. There's a certain probability that it will happen, and 1 minus this probability that it won't. Now in this expression, this is the diffusion coefficient of our particles, and kappa is the reactivity of their encounter, which is a constant related to the probability that our guys can actually bind when they're close enough. The DNA walkers that were used in these experiments, they're quite small, and hence they're moving very rapidly. So diffusion is fast, we can neglect the leftmost term in this expression, and binding is then reaction limited. So if we define A as the distance between the footholds of the walkers, we can obtain a communication speed by dividing this distance by the mean binding time. And not surprisingly, the higher the probability of a successful binding, the higher kappa, the higher the communication speed. And the higher the probability of finding the walkers at a distance where they can bind at epsilon, the faster the communication speed as well. So from our analytical expressions for Q, and using a model for kappa derived by Eric Winfrey's group, we estimated the communication speed of the DNA acrobats, which was found to be in very good agreement with experiments. But the most important thing is that we actually got a maximum, an optimal length of the molecules that's going to lead to the maximum velocity for a given separation. So you can see here that we got a velocity of about 7 nanometers per second. So that's actually the fastest velocity that has been observed to this date. But this is only in theory. Having all the walkers moving around randomly in a 2D array, not in a linear reversible way, does not transport signal at its maximum speed. We will actually get a velocity that's about 20 times lower with all this back and forth. So how can we optimize this? How can we fully benefit from this high-speed communication? It's evident that we must put all these walkers in a row and make motion irreversible. Then we must optimize the mechanism of communication. And we analyze 4 different mechanisms of communication. One where one walker diffuses in 1D towards a fixed target, and binding to its target is going to trigger the motion of the next walker. One where the 2 walkers diffuse in 1D, and the signal is transmitted from one to the other when they are in close contact, and the same mechanisms but in 2D. Then, deriving expressions for the mean binding time for all these mechanisms, we observe that having 2 walkers diffusing in 2D is the less efficient of all these methods, as the walkers sample a lot of phase space before their encounters. And the optimal communication speed is found when entropy is reduced the most, when a single walker reaches a fixed aim in 1D, triggering the motion of the next one. So with this in mind, we've been working with our experimental collaborators from Ohio State in 2 things. First, we're working designing DNA origamis that allow for reversible motion. We're also working on designing DNA origamis that allow for 1D communication. And I'll walk you through our advances in these 2 fronts. So to study reversible motion, we started working with a device that uses steric interactions to forbid the signal from going back. So notice that when the left arm is closed, the right arm has to remain open and the other way around. As a first effort, we're working with just 2 pairs of interacting arms, and the motion of these arms is characterized by the behavior of the hinges, the stiffer the hinge, the more close the arm is, and by the overhangs that we place in the arm and in the base. So the more overhangs we have on one arm, the more likely it is for this arm to be found closed. It's going to stay closed for longer times, and the stronger the overhangs, the same is expected. So to characterize the dynamics of these interacting arms, to see how one arm influences the dynamics of the other one, we perform Brownian dynamic simulations of this device that you see over here, which were informed by the probability distributions of isolated single-arm dynamics. So we impose energy penalties on the hinges that model their experimental distributions, just like we see over here, and we associated binding and unbinding probabilities to the overhangs to keep the arms down, which we assigned so as to match single-arm measurements. We simulated a lot of these devices, compared our results to experiments, and found excellent agreement. That is, this simple model accurately predicts how the arms interact, and then behave just as expected. So next we plan to design a similar device that will propagate a signal over large length scales. But the junctions here may be hard to control. So we've been simultaneously working with a different setup. We've been playing with this idea of 1D communication on a set of V-shaped structures designed to move on a line. And our collaborators are currently taking Fred measurements to characterize the kinetics experimentally. So once we know the kinetics of an individual device, capturing the multi-device kinetics is trivial. We have analytical expressions for propagation time for these reversible systems, and we can also perform simulations just like the ones we previously showed to validate this. So I think this is pretty interesting because we can amplify a single binding event that will trigger this motion just as in a domino, and these kind of binding events can be effectively used for biosensing applications. So in summary, using 1D motion and making communication irreversible has the potential to lead to the fastest communication speeds in the nanotechnology, at least in theory, and we developed theories to estimate these communication speeds. We're also currently working on designs that allow for these motions to develop such fast communications. So thanks for your attention. We're running out of time, so feel free to send me questions or comments at ss1212@duke.edu.